Good evening, students. Due to some technical problems, we could not continue the botany session yesterday. Today again, we'll start from the beginning, follow it. The diversity of living organisms. The organisms means the plants and animals. Plants and animals are called animals. Now, the main subject, biology, study of living organisms, it is differentiated into botany and zoology. Botany, the study of plants, is also known as phytology. Phyton means plants, ology means study of plants, phytology. Next, zoology includes the study of animals. Now let us consider the plants. So far it is estimated that 3,43,000 plants. It's very difficult to study all the plants in individual, so that it should be divided into groups and subgroups, etc. In that way, many biologists classified plants in different ways based on the characters they consider. Among these, the old classification by A.V.V.S. 1883, he classified the plants as follows. First, he kept all the plants in one kingdom, that is plant kingdom, and he classified the plants into two groups based on flowering. The first group, the cryptogams. Cryptos means invisible, gamos means sex organ. Invisible sex organ plants, also called non-flowering plants. Second group, phanerogams, means the flowering plant. Now let us consider the cryptogams. These cryptogams are classified based on the plant structure, or thallus structure, or plant body structure. The first one, thallophyta. Thallophyta means thallus having plants. What is thallus? Thallus is a plant body that we cannot differentiate into root, stem and leaves. That is called the thallus. Now let us consider these thallophytes. Thallophytes are again classified into algae and fungi based on the nutrition. The algae plants are having chloroplast, so they can prepare their own food material, so those are called autotrophs. Whereas the fungi do not have any chloroplast, so they depend upon other organisms for their nutrition. That is called heterotrophic nutrition. This heterotrophic nutrition may be saprophytic, parasitic or symbiotic. Now let us see the algae. As I told you already, they are having chloroplast. Majority of the algae are aquatic. They live in water, and they have a thallus organization ranges from unicellular to multicellular forms. And then, as I told you already, they cannot differentiate the stem, root and leaves, etc. And another important thing, the cell wall is made up of cellulose, and whereas here in fungi, the cell wall is made up of chitin. Here the reserve food material is a starch, and here glycogen and oil drops. And in both the sex organs are unicellular, not having any cover cells, any jacket cells. And in both, after sexual reproduction, no embryo is formed. So these are called non-embryophytes. Plants do not have any. So let us see some examples. Note down. Chlorella, Chlamydomonas, Pyrogyra, Ulothrix, Laminaria, Nostoc, Anabina are some of the examples. Many plants are there in algae. These are some examples to know. Regarding fungi, Rhizopus, yeast, Penicillium, Agaricus, Cercospora, Mucor, Aspergillus. Many plants are there here also. Now, next go to Bryophyta. Bryophyta, this is a plant group, and these plants are the first land plants, and they're having, instead of roots, they're having a rhizoid structure so that they can absorb water from the ground. Another important thing, they're also having chloroplast, or they're also autotrophs, and reproductive structures having sterile dockets. Important point here. And another thing, these bryophytes, the dominant plant, the plant that which we can see is a gametophyte. Gametophyte, remember, gametophyte is always haploid and sporophyte is always diploid. And this plant also having two phases in its life, gametophytic generation and sporophytic generation. Gametophytic generation having sex organs, Antheridia and Archegonia. Antheridia gives male gametes, Archegonia gives female gametes. After pollination and after fertilization, they form into zygote. Zygote is diploid and it gives the diploid sporophyte. Sporophyte having sporangium and it is having spore mother cells. They undergo reduction division and form haploid spores. Haploid spores again develop into bryophyte. This is called alternation of generation. So this is a land plant. At the time of reproduction or fertilization, it needs water. So that this is a amphibious plant. Bryophytes are called amphibians of the plant kingdom. In this, it is also again, Bryophyta is also again classified into this Anthoceratopsida, Bryopsida and Hepaticopsida. In this, Trixia, Marcantia, Anthoceras, Funaria are the examples for this. Next, we'll go to Iridophyta. Iridophyta, Iron. Iron means feather, feather-like. Iron means feather-like. Feather-like means in a feather, you will happen to see there is a central vein is there. Central vein is there, and on the central vein we have the leaves are like this. Right, leaves are like this. So these are called. This is also called the fronds. The leaves of pteridophytes is called the fronds, where the leaves are like this. So it is called a Pteridophyta. Pteridophyta, this study is pterology. Study of Pteridophyta, pterology. Study of Bryophyta, bryology. Study of algae, phycology. Study of fungi, mycology or mycetology. So now, this pteridophyte characters. This is a dominant phase. Here also you can find the sporophytic generation and gametophytic generation. The sporophytic generation is a diploid plant, diploid, and gametophyte as usually haploid. This plant having the root, stem, and leaves just developed. And another important thing here, these plants having vascular tissue. Vascular tissue includes xylem and phloem. So these are the first plants having xylem and phloem. So these are called vascular cryptogams. The pteridophyta are called the vascular cryptogams having the vascular structure. And so for in xylem, algae and fungi, no embryo is formed. There is in bryophyta, after gametic fusion, embryo is there. So this is an embryo phyte. Embryophytes are plants having embryo after gametic fusion starts from bryophyta. Bryophyta, pteridophyta, gymnosperms and angiosperms are called embryophytes. And now in the pteridophyta, another characters, the leaf is known as the frond, and it is having the sporangia on the lower side of the leaf, ventral side of the leaf. So what happens? These are called spore-bearing leaves are called sporophylls. Sporophylls means spore-bearing leaves are called the sporophylls. So this plant produces the sporangium. The sporangium, since it is diploid, it undergoes reduction division and form haploid spores. Haploid spores are dispersed out and germinate into a gametophyte. Gametophyte is haploid. Generally, the gametophyte of pteridophytes are called prothallus. Gametophyte of these pteridophytes, prothallus. And then this prothallus having an androecium and gynoecium, and they give male gametes and female gametes. After gametic fusion, a diploid zygote is formed. Zygote directly develop into sporophyte generation. So in this both you can have this alternation of generation. Alternation of generations of sporophyte and gametophyte, like that. Since I told you already the vascular cryptogams, and these are the first plants where you can find the xylem and phloem is developed. Xylem and phloem developed in pteridophyte. Here are the examples: Lycopodium, Selaginella, Neprolepis, Equisetum, Polypodium, etc. Many plants are there in pteridophytes also. Next, gymnosperms. Here, these are gymnosperms. Gymno means where the ovule is not protected. Ovule is not protected, so those are called the gymnosperms. And highly developed plants, well developed root system, stem and leaves. It produces the cones instead of flowers. Produces the cones. And gymnosperm is a sporophyte, a diploid plant. It produces the male cone and female cone, etc. And male cone produces the microsporophylls, microsporangia, and female ovule you can find here. And after pollination fertilization, it gives zygote. Zygote develop into embryo. Zygote develop into embryo, and embryo will give again the diploid gymnosperm. But here the difference is the vascular system so developed here. You can find xylem is restricted to the tracheids only. Xylem is restricted to tracheids, and phloem sieve cells not highly developed. And here some examples: Pinus, Cycas, Netum. When coming to the angiosperms, the angiospermos, these sex organs or ovules are enclosed ovule plants. And also these root, stem and leaves are highly developed. Maximum taproot system you can find, and then monocots you have this fibrous root system you can find. And then depend upon these cotyledons, they are classified into two groups: monocotyledons and dicotyledons. In an embryo, if there is a single cotyledon, those are called monocotyledon. The single cotyledon is also called the scutellum. Single cotyledon of monocots is called scutellum. In dicots, there are two cotyledons here. And here, highly developed xylem. There you can find xylem is having the tracheids and the vessels. Phloem is having the sieve cells, sieve tubes, companion cells, and phloem parenchyma and phloem fibers. Whereas in this one, regarding the examples of monocotyledons, many plants here you can have this monoplants. In monocots, the examples of paddy, wheat, and maize. Paddy, wheat, and maize. And regarding these dicots, hibiscus, mango, neem, that is available in our areas. There you can find monocots, and then many differences are there in this. Regarding the monocots, there you can have fibrous root system, that is a type of adventitious root system, and the leaves having parallel venation, and the flowers are trimerous, whereas in dicots is having taproot system, and the leaves are having reticulate venation, and flowers may be tetramerous or pentamerous condition we can find in this. So that is the main differences between the monocots and dicots. Remaining thing we can discuss in the chapter taxonomy. So now the next classification given by Linnaeus in 1753. It is a two kingdom classification. The primitive type, where he classified all the living organisms into two large kingdoms based on the type of nutrition. That is only main here. The kingdom Plantae, there you can find all the plants. They are autotrophs. Cell wall is made up of cellulose, and the reserve food material is starch. Whereas the second kingdom, Animalia, these are all animal kingdoms. They are moving from one place to another place, and no cell wall, there you can find, and they are maybe heterotrophs. So based on this only, we classified these two plants. This is only for historical interest, we can follow this Linnaeus system of classification. Next, the next classification, that is called a three kingdom classification. It is proposed by Haeckel in 1894. So in addition to this Plantae and Animalia, he introduced another group that is called Protista, where he included the unicellular organisms including protozoans, algae, fungi, bacteria, and slime molds, mild molds. And then Plantae included all multicellular plants, and Animalia multicellular animals. It's also got any demerits in this, because only simply having the unicellular organism are separated in a separate Protista group. Next classification is somewhat a better classification than this previous one proposed by Whittaker in 1969. He included into five kingdoms, which is Monera, Protista, fungi, Plantae, and Animalia. Now let us see what are the criteria he has taken in classification of this classification. So the criteria for this classification is based on the body structure. Has to take the first point he has taken. The body structure, that is the complexity of the body structure, that is from unicellular to multicellular structures. And then the second one, the complexity of the cell structure, prokaryote to eukaryotes. Another condition is has taken the mode of nutrition, that is autotrophic or heterotrophic nutrition, and then the lifestyle. They may be predators or consumers or decomposers. Based on this, he classified the plants. The first he had taken the Monera. Monera, he took prokaryotic organisms. What are prokaryotes? Prokaryote. Protos means primitive, karyo means nucleus. The organisms having primitive type of nucleus. What are the primitive characters? Nucleus, not have nuclear membrane, and the absence of mitochondria, chloroplast, Golgi bodies, and endoplasmic reticulum. Those are called the prokaryotes. We include mycoplasma. This is smaller than the bacteria and causes diseases. Another is the bacteria. So you can learn later regarding the bacteria. And actinomycetes, this type of bacteria, the filamentous bacteria, and the cyanobacteria, that is blue-green algae. Blue-green algae are named as cyanobacteria. And the second one, the protista, is protista and third kingdom. Three kingdom protista is different. In this included unicellular eukaryotes. Another term, eukaryote, I told you already regarding the prokaryote. Eukaryotes are advanced, having advanced nucleus. What is advanced nucleus? Having definite nuclear membrane and having chloroplast, mitochondria, cytoplasmic reticulum, and Golgi bodies. If you go deeper, there you can find the ribosomes are of 80S. Here you can find only 70S type of ribosome. And mostly aquatic plants, eukaryotic cells, unicellular eukaryotes included in the protist. And the next is the fungi. These are heterotrophs, includes eukaryotic, unicellular or multicellular. So except prokaryote included all fungi in a fungi group, a separate group, there you can find eukaryotic, unicellular and multicellular plants. They're all heterotrophs and decomposers. Heterotrophs and decomposers. Regarding the next group, Plantae included multicellular plants. This is separated unicellular. It included all multicellular plants, which include algae, bryophyta, pteridophyta, gymnosperms, and angiosperms. Included all this Plantae. And the next one is Animalia. There included all the multicellular eukaryotic animals. Multicellular eukaryotic animals. To this, you know already the differences between the plants and animals. So this is the classification. There, some merits and demerits also there. Take into account some characters because cannot deny the classifications of the high people, biologists. So they take into account some characters in their mind and used to classify the plant. But other scientists used to take the other characters and they used to classify the plants. So that we can compare these things, but you cannot hesitate to take the classifications of these people. So this way, that is the diversity, the plants diverse into different groups depending upon the characters they have. Please wait some time. We'll continue the next lesson. We come to the next chapter. Viruses. Viruses means poison in Latin because they cause diseases. That's why they are called poisonous viruses. To understand virus, here is a definition. Viruses are defined as non-cellular. Non-cellular means we cannot consider it as a cell. A cell means it's having a plant cell having a cell wall, plasma membrane, nucleus, and cell organ. There is here you don't find protein layer, we have only a nucleic acid. So they are not considered as a cells. So it's a non-cellular. And ultra microscopic means not see with our naked eyes. I think you know 1 micron is equal to 1 by 1000 millimeter. 1 by 1000 micron is equal to millimicron. So you can see we can these viruses in millimicrons. So that you can see only the electron microscope only, not with normal microscope. And infectious, that is, they infect people or they infect organisms, both plants and animals, and cause this. Intracellular, they are inside the cell. When there is outside the cell, they are not living, they cannot do anything, they are not infectious. And when they are inside the cell only, they will be causing diseases. Obligate parasite, that is, Definitely parasite only. There is no alternate. So some fungi, if you happen to see, they are saprophytes, sometimes parasites. The parasites may be a saprophyte. So not like that. They are obligate parasites means strictly they are parasites. And they are made up of nucleic acid. The nucleic acid is known as the nucleoid. Nucleoid means like. Nucleus like structure, so it is nucleoid. And this nucleic acid that is surrounded by the protein coat. So here you can have a nucleic acid. This is surrounded by protein coat. The protein coat is made up of number of protein subunits. Number of protein subunits. The protein subunits are called capsomers. They are called the capsomers, and in the center you have this nucleoid. Recording the history, see the viruses are not at a time discovered, at different places they are discovered. So the discovery is very interesting. Either bacteria or viruses. The first one is Edward Jenner in 1796 controlled smallpox virus disease through vaccination. Actually, at the time, virus was not discovered. But as a physician, he is a doctor, as a physician, he controlled this disease through vaccine. He prepared a vaccine and controlled the disease. And then Adolf Meyer, 1886, studied tobacco mosaic disease and demonstrated that the disease is transmissible and infectious. He observed the tobacco mosaic disease and collected the sap from the plant, and he applied the sap to another tobacco plant and find out that that plant having disease. So this is transmissible. It may be transmissible from one plant to another plant. And Dmitry Ivanovsky actually is the person who named a virus. He discovered viruses and demonstrated that the casual organism could pass through bacterial filter. At the time, to filter the bacteria, filters are available. So he used that filter and infiltrate is given to another plant, the same disease came. So that this organism is not filterable. And these filters, bacterial filters, are prepared by Chamberlain, discovered the bacterial filters in 1884. And Hezrink in 1898 named this infectious fluid, that is the sap coming from the infected plant, infecting another plant. So he called it as a contagium vivum fluid. So it is a living infectious fluid. So it causes disease. Even the fluid after the infiltration also causes the disease. Next, Lofer and Froch, 1898, discovered viral diseases in animals. Mott, 1915, discovered the first viral disease of bacteria. Next, D. Harrelly coined the term bacteriophage. What is a bacteriophage? A virus infecting bacteria is known as bacteriophage. And W. M. Stanley, 1935, crystallized tobacco mosaic virus. Crystallize means make into powder. So we can call it as a non-living organ, non-living things. Because living things cannot crystallizable, so this is non-living. Retain the infectivity after a long time. Even that powder also causes disease. Even after crystallizing the tobacco mosaic virus, and that crystals cause disease, and they retain the infectivity for nearly a long time. And Bowden and Perry established nucleoproteinaceous nature of viruses. As I told you already, the virus, there is a nucleoid or nucleic acid surrounded by a protein coat. This protein coat that is proposed by this Bowden and Perry, established nucleoproteinaceous nature of the virus, nucleus and protein. And next, Ginder and Linderberg. Linder and Ginderberg discovered the ultrastructure of tobacco mosaic virus. He studied the structure and discovered the structure that we'll learn later, we'll discuss later, proposed by this Franklin et al. In 1957. Lindman, 1957, successful in vaccination against polio. He prepared this polio vaccine. And Isaacs and Lindman discovered interferons. So these interferons are the proteinaceous materials that are developed in an infected virus by the animal or in human beings. They develop these interferons inside when you are infected by these viruses. Next, Sinden discovered mycophages. Mycophages are viruses which infect fungi. And Schafferman and Morris discovered cyanophages. Cyanophages means the viruses which infect the cyanobacteria or blue-green algae are known as cyanophages. And then Steiner and Reimer discovered viroids. Viroids is, as I told you already, a virus having a protein coat and a nucleoid and infecting. Whereas these viroids, a protein-free and this RNA, two-stranded, double-stranded RNA, it is having only the protein coat, discovered protein-free RNA, only the nucleic acid without protein coat causes the disease. And Robert Gallo discovered AIDS, that is acquired immunodeficiency syndrome, that we will discuss later. And Prusner, 1997, discovered prions. These prions are infected to proteins. Only protein causes disease. No nucleic acid, only the protein causes the disease. Those are called the prions. Okay. So now let us see. As I told you already, this may be crystallizable, etc. To explain, it is not a cell but infectious and ultra microscopic, obligate parasite, etc. Those are the characteristics. So now let us see this having both living and a non-living character. Let us see the living characters of viruses. So living characters, the ability to multiply inside the host. When a virus enters into the host, it multiplies and giving many bacteriophages or many cells of virus. Property of mutation. Mutation are the sudden changes due to the temperature or this cold or by chemicals, etc. That is a mutation, but these viruses are having this mutation property. Next, having nucleic acid and proteins. As I told you already, there is a nucleic acid is there that is surrounded by protein coat. So both are living characters. And react to heat, chemicals and radiation. So if a virus is exposed to a radiation not here, it is just pass away, move away. So those are the characters of the living characters of this virus. And then non-living characters. So what are the non-living characters? Inactive outside the host. It is not outside the host, in outside the atmosphere, it is non-living, cannot do anything. And have crystallizable, I told you already, Stanley crystallized these viruses into powder. So that nature is a non-living nature. And absence of any metabolic activity outside the host, no metabolic activity, no growth, no reproduction, no respiration, etc. And cell wall, cell membrane of any type is absent. You don't find any cell wall here. And no plasma membrane. Wall is animal character, no cell wall. And cell membrane having plant and animal character, that is also absent. and no protoplasm inside the virus. So these viruses we cannot consider as non-living, cannot consider as living. It is between living and non-living organism. Now let us see some unique characters of viruses. So having living characters, non-living characters, these are the unique characters that you never find any plant group. Include the presence of only one type of nucleic acid. Generally in all organisms having both RNA and DNA, but whereas in viruses we are having only one type of nucleic acid. Maybe either RNA or DNA, not both. Next, capacity to reproduce from their own nucleic acid. For example, if any reproduction is there, reproduction it involves both nucleus, cytoplasm, and other cell organelles. But whereas here the sole nucleic acid continue the reproduction activity. Next, to make use of the ribosomes of the host cells, since this virus do not have any ribosomes, so whenever it is infected to the host, it makes use of the ribosomes of the host cell for the preparation of proteins. And next, so that's why Prof et al. 1966 consider the humorously the viruses are viruses because these are the unique characters that you can find only in viruses, so it's called the viruses or viruses. Next, let us see the classification of viruses. The classification of viruses, there are some criteria to classify the viruses, that is the type of nucleic acid, whether having RNA or DNA, and symmetry of the particle, and then the presence or absence of envelope around the nucleocapsid. So, this virus is having envelope around this nucleocapsid. So, based on this, the viruses are classified. So, all the viruses are kept in one phylum, phylum A.V.V.S. It is subdivided into subphylum Deoxyribonucleic, where the nucleic acid is deoxyribonucleic acid or DNA. The second one, subphylum Ribovira, it includes the nucleic acid, the ribonucleic acid, RNA. So again, further classified into this, three. This deoxyvira, one is deoxycubica. So there you can find RNA is the genetic material, and the shape is cubical shape, deoxyhelica, helical shape, deoxybinella, binal shape. In subphylum Ribovira, again classified into two classes. Ribohelica, this is having RNA and helical nature. Nucleic acid is RNA, this is cubical nature. So in the same way, the viruses are classified based on some criteria only. And now let us see actual the structure of these viruses. So before that, let us see the size of the viruses. Viruses varies from 10 to 300 nanometers size. The smallest animal virus, 10 nanometer diameter, that is foot and mouth virus. And the smallest plant virus, that is 17 nm diameter, example satellite virus. And the largest animal virus, that is nearly 250 nanometers, example pox virus. And the largest plant virus, 300 nanometers, that is tobacco mosaic virus. So regarding the plants, the smallest plant virus is a satellite virus, and the largest plant virus, that is the tobacco mosaic virus. Regarding the shape, it is divided into 5 types. The spherical shape, the influenza virus, the spherical shape, and the rod shape tobacco mosaic virus, rod shape, and a tadpole shape that you can find in a bacteriophage, and brick shaped pox virus, And adenovirus, that is polyhedral. Many angles you can find, that is polyhedral virus. So now let us see the structure of tobacco mosaic virus. It's common in tobacco plantation. This one is a rod-shaped virus. The rod-shaped virus, and here is a central. Here you can find a single-stranded RNA is found in the center, has a spiral manner, and around this you can have a protein coat. This protein coat having subunits, protein subunit is called the capsomeres. And this one here you can find the total diameter 18 nanometer diameter and 300 nanometers length. This is tobacco mosaic virus. Next we'll see the structure of the bacteriophage. I told you already the bacteriophage is virus infecting bacteria. It is a tadpole shape, having a head and a collar and a tail piece. Three parts will have. In this head, it is hexagonal and having a double-stranded DNA inside, and there is a collar because there is a contractile sheath in the center, and at the end you can have basal plate. This basal plate having 6 tail fibers and 6 prongs. These tail fibers and prongs help to sit on the bacterium, and during this multiplication, it sends this contractile sheath contracts and send this DNA to the what's called the parasite, the host cell, and then the replication takes place. They're all moving in the atmosphere. These bacteriophages, a number of bacteriophages which are moving in the atmosphere. So tomorrow we'll study the multiplication phase of this bacterium. Thank you.